Welcome to the second part of our discussion. To review, when immunogens are encountered, there will be a corresponding immune response. Antibody production will result following activation of B cells, or there will be cell death as mediated by cytotoxic T cells. The question now is, what determines the immunogenicity of a molecule? And how do these factors relate to the immune response? On the succeeding slides of our discussion, I would like to refer antigens and immunogens as the same, so please do not be confused. Now, these are the different factors that determine the antigenicity or immunogenicity of a certain molecule. First is foreignness. Foreignness refers to the degree of which antigenic determinants are recognized as non-self by an individual's immune system. Remember that your immune system is normally programmed to discriminate self and non-self antigens. Self antigens are tolerated, hence they are not attacked by the immune system. Only the non-self antigens are attacked and eliminated out the body. An antigen must be foreign, alien, or non-self to which it is administered. The degree of the foreignness of an antigen influences the amount of immune response elicited. Therefore, the more foreign an antigen is, the more it will be recognized by your immune system as non-self, and a corresponding response will be activated. The more distant taxonomically the source of the immunogen is from the host, the better it is as a stimulus. Now, there are different degrees of foreignness that our immune system is able to recognize antigens as non-self. First is autologous antigens. Example, Maria Clara decided to bank her whole blood doon sa Red Cross. Then Maria Clara suddenly met an accident. The whole blood the whole blood that Maria Clara banked could be used to transfuse back to Maria Clara. And it is expected that no adverse reactions will be encountered. Why? Because the whole blood contains autologous antigens. Autologous antigens come from the same individuals. Another antigen is we call syngenic antigens. Syngenic antigens are those that come from twins. Twins who have the same genetic makeup, as in identical twins. Another type of antigen according to foreignness is allogenic or homologous antigens. These are antigens that come from different individuals from the same species. Example, kidney transplantation between siblings. Okay? Another is synogenic or heterologous antigens, as we have discussed earlier. These kind of antigens are antigens that come from another species. And the example I gave in the previous video on tilapia skin grafts used for burnt patients. Again, the rule of thumb, the more foreign the molecule is, the more it will be recognized by the immune system as immunogenic. However, there are instances that cross-reactivity occur. Cross-reactivity occurs wherein one antibody directed against one specific antigen is also able to interact with another different antigen. This may be because of similarities in their molecular structures. Okay? Take a look at this illustration here. This antibody is directed against this antigen. This antigen looks like another antigen. Yung difference nila is just a little, but the antibody is still able to recognize such antigen. So we call this as cross-reactive antibodies. Typical example is the post-streptococcal sequela rheumatic fever. An infected person produces anti-streptococcal antibodies. These antibodies either are directed against streptococcal antigens, but to some degree, our myocardium has structures similar to the M protein of Streptococcus pyogenes. And because of this, the antibodies are able to cross-react with our myocardium, causing damage. Another factor is degradability. Degradability pertains to the property of the antigen to be processed by MHC and to be presented by antigen-presenting cells. For a substance to elicit an immune response, it must be subjected to antigen processing. Antigen processing involves digestion of the antigen in order to create small peptides. Now, these small molecules or small peptides will be complexed with MHC molecules. And this complexing will be expressed on the surface of the antigen-presenting cells. And the antigen-presenting cells will present the antigen to the lymphocytes, particularly the T lymphocytes. Now, when an antigen is presented to T lymphocytes, a stronger immune reaction is expected. Remember in our discussion in the previous video, a T cell dependent antigen can provoke a stronger immune reaction. Likewise, a T cell dependent antigen can cause immunologic memory. So it is really expected that when an antigen undergoes antigen processing and presentation to T cells, there will be a stronger immune reaction. If a macromolecule cannot be degraded and cannot be processed with MHC molecules, therefore it is considered as poor immunogens. Next is molecular weight. The rule of thumb is that the higher the molecular weight, the better the molecule will act as an antigen. Molecules having molecular weight below 5,000 Daltons are considered non-immunogenic, whereas those antigens having higher molecular weights, such as albumin with 40,000 Daltons, gamma globulins 160,000 Daltons, hemocyanin 1 million Daltons. These are big molecules and they are therefore considered as excellent immunogens. But does size really matter? Yes, ako na mismo magsasabi sa inyo, size matters because the number of epitopes increases proportionately with the size of the molecule. As you can see here, the smaller size has fewer epitopes, whereas the larger size have more epitopes. And with that, we can say that truly molecules with higher molecular weights, molecules that are larger in size, are good immunogens. And aside from that also class, larger molecules are more immunogenic because they are easily phagocytized. And likewise, antibody formation is increased if the antigen undergoes antigen processing. And larger molecules are more likely to undergo antigen processing. Now, when it comes to molecular weight, we may also encounter haptens. Haptens are actually low molecular weight proteins which by itself cannot elicit formation of antibodies, but they can bind with an antibody. Haptens therefore are antigenic, but they are not immunogenic because of their size. They are too small to elicit an immune response. Therefore, a hapten must be coupled with a carrier molecule. The carrier molecule will increase the size of the hapten, and when such happens, the haptens can now become immunogenic. Example of carrier molecules for haptens are albumin or globulins, okay, and other synthetic polypeptides. Another factor is structural stability. The strength and solidity of a molecule determines its efficacy as an antigen. The more solid the molecule, the stronger the molecule is, therefore the more stable it is, and therefore the more immunogenic it is. Now, unstable molecules such as gelatin, they are considered poor antigen. Remember, gelatin can become solid and can also become liquid. So, unstable kanyang molecular structure. Likewise, static molecules such as inert molecules are also poor antigens. Stable and mobile antigens are important if the goal is to induce an immune reaction. And this is true for vaccines because vaccines must contain antigens that are stable and at the same time mobile in order to induce a stronger immune reaction. Now, another factor is the chemical nature of the molecule. As we have mentioned earlier, antigens can be large organic molecules. They can be proteins. Proteins, well, majority of immunogens are proteins because proteins are the strongest immunogens because of the complexity of the structure of proteins as well as the molecular weight. Proteins are basically heavy molecules. They are large molecules. Second, glycoproteins. These are best illustrated by RBC blood group antigens and MHC antigens. Your blood type A antigens, type B antigens, those are made up of glycoproteins. Another is polysaccharides. Polysaccharides are mostly haptens. They are too small to be immunogenic. They are also referred as incomplete immunogens. Polysaccharides also does not possess sufficient chemical diversity kasi nga repeating molecules of CH and O ang polysaccharides. So with that, we can say that it's a poor immunogen. However, and likewise, they are also easily degraded. But when polysaccharides are coupled with proteins, they become immunogenic. For example, glycoproteins. Glycoproteins are made up of a polysaccharide and a protein. Another is nucleic acids. Nucleic acids are basically non-immunogenic because of the simplicity of their structure, except for single-stranded DNA. Nucleic acids are also flexible and they are degraded rapidly, so they are unstable molecules. But when nucleic acids are coupled with proteins, for example, they become immunogens. The proteins stabilize the overall structure of the molecule and thus becomes immunogenic. Another is lipids. Lipids are basically non-immunogenic except for cardiolipin. Lipids are non-immunogenic because they are relatively simple and they also lack structural stability. But when they are linked with proteins, they become lipoproteins and therefore they can now be rendered immunogenic. Another, we have polypeptides such as hormones. They are referred as weak antigens. Okay. Adjuvants. Now, adjuvants are referred as the dirty little secret of vaccines. Adjuvants are immunopotentiating agents, meaning they enhance the immune response against a certain immunogen. The vast majority of modern vaccines do not work unless they contain adjuvants. For safety reasons, modern viral vaccines such as hepatitis B, they only use part of the virus and not the entire virus. And because only a part of the virus is used, that part of the virus is not that stable. It's not a very good immunogen, and that is why that kind of vaccine requires the presence of adjuvants. The functions of adjuvants include: first, they increase antibody production. They decrease the threshold dose of antigen. They do not require large dose of antigen to become immunogenic. And they also make weak antigens to become potent and strong. Now again, without adjuvants, uh, vaccines such as that of the hepatitis B is doomed to fail. Now, what is the mechanism of action of the adjuvants? Adjuvants give a slow release of antigen adjuvant complex to the immune cells. So, dahan-dahang ini-introduce ang antigen to the immune system. Likewise, adjuvants can also increase the size of the antigen, and so they become a good immunogen. And adjuvants also increase the efficiency of antigen presenting cells and they increases the lymphokine production. So with that being said, we can say that the reason why vaccines work that way, that they are able to effectively provoke our immune system to elicit a corresponding immune reaction, is because of the help of the adjuvants. Adjuvants are immunopotentiating agents. They help to enhance the immune response against a certain immunogen. Example of adjuvants, we have water and oil. Alum precipitate, by the way, is the most common adjuvants used in modern vaccines. We also have synthetic polynucleotides. We have bacterial endotoxins. And the Freon's complete adjuvant. Freon's complete adjuvant consists of killed Mycobacterium tuberculosis and oil in water emulsion. And the action of Freon's complete adjuvant is it increases the cell-mediated response. And it also increases antibody production. So, 'di ba? It enhances the immune reaction by activating both the cell-mediated response and humoral response. So, that ends our discussion for antigens. I hope that you learned something from me. And if you have questions, kindly post them to Edmodo or to our Telegram. Thank you so much for your time and for listening.